Hello everyone, welcome to our channel. Today's subject is food chemistry and chapter is carbohydrates part 3. I am Srijita Ghosh and I am your tutor for today's lecture. So let's start. Structure of glucose. There are different forms of cyclic forms for sugars and Fischer projection for alpha D glucose. We can see the picture over here. This is D glucose and alpha D glucose. This is the cyclic form and beta D glucose cyclic form. Fischer's and Howard's projection. Fischer's projection is cyclization of glucose to hemiacetal is spontaneous to form stable ring structures. Howard's projection, glucopyranose 5-membered ring with oxygen atom in it and glucopyranose is 6-membered ring with oxygen atom in it. Monosaccharide can cyclize to form pyranose or furanose form. See, this is D-glucose. It can be cyclized to form pyranose form like this and also it can be cyclized to form furanose form like this. These are the images of chair conformation of glucose. This is alpha D glucopyranose and beta D glucopyranose. Occurrence of sources of glucose. Glucose is found in free or bound state in nature. Glucose is a component of disaccharides and polysaccharides. Glucose found in foods, human blood and body cells. Physical properties of glucose. Glucose possesses asymmetric chiral carbon atoms in its structure. This confers two physical properties: optical activity, stereoisomers. Optical activity of glucose. Optical activity for an aqueous solution of glucose is dextrorotated. This is denoted by +D. It rotates a plane of plane polarized light in polarimeter towards right. Hence, glucose is also termed as dextrose. Specific rotation of glucose and optical activity. Pure alpha D glucose has a specific rotation of plus 112.2 degree and pure beta D glucose has a specific rotation of plus 18.7 degrees. Functional or structural isomers of glucose. Glucose, also known as aldohexose, and fructose, also known as ketohexose. These are functional isomers. Their structure differs only at functional groups, that is either at keto group or at aldo group. Glucose stereoisomers. There are three different types: D and L glucose, anomers of glucose, and epimers of glucose. See, this is the D glucose and this is the L glucose. Only the changes is in the OH form at the C5 position of the glucose. This is a mirror and this is non-superimposable mirror images and known as enantiomers. Physical and chemical properties of enantiomers are same except optical rotation. Sugars present in human body are of D series. Glucose anomers of Howard's projection shows as follows. Alpha glucose has OH group below the plane and beta glucose has OH group above the plane. See, in alpha D glucose the OH is below the plane and in beta D glucose the OH is above the plane. Epimers. Glucose has two epimers. One is mannose and another one is galactose. What is the difference from glucose to mannose? In the C2 position, there is change, and similarly in galactose, in the C4 position, there is a change with the glucose. Now see, in case of mannose, in C2 position, this is C2, the H and the OH are at different sites. Similarly, in galactose, at the C4 position, this one, the H and the OH are at different directions. So, these are epimers. Mutarotation. Mutarotation is change in specific rotation of an optically active substance. Mutarotation is a physicochemical property shown by certain optically active substances who in aqueous solution has capacity to interconvert from one stereoisomeric form to other and attain a constant equilibrium mixture. This change This changes an initial specific rotation to a constant specific rotation with the passage of time. Thank you. Thank you for watching our video. If you like the video, please hit the like button and subscribe to our channel. And please share with your friends. Thank you once again.